This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 289, recorded on June 13th, 2014. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses, coming to you on Friday the 13th. Ooh, scary, huh? Joining me today in the TWiV studio, an unfrightened Dickson Despommier.
Yes, I'm steadfastly unfrightened.
Did you know it was the 13th?
I did.
Of Friday?
I did.
And it didn't bother you?
Not yet.
All right.
The day isn't over.
Yes, we have a long ways to go. So joining us today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Is it Friday the 13th over there?
It is, and here in Ann Arbor it's 67 degrees and partly cloudy, partly sunny.
Wow.
Partly cloudy, partly sunny. So is your cup half full or half empty?
Oh, it's half full.
Cool.
Did it rain just before it became partly sunny?
No, it hasn't rained today at all.
Because this morning it was so densely fogged where I live that I was afraid to actually drive to work.
Wow.
Yeah, it was quite foggy. And now it's 22°C and partly cloudy.
Or partly sunny.
Partly sunny. Yeah, it is quite nice out there over the Hudson River.
A little humid.
Quite. And the air conditioner is broken here in the Hammer building. Right?
Exactly right.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
You know that broken Hammer air conditioning story, right, Alan?
Oh, I have lived it.
It's not a story.
We managed to open some of the windows at one point.
Really?
They probably fixed this, but I think with a screwdriver and an Allen wrench, you used to be able to kind of open the window a few inches, and then of course, since you're 13 floors up, you're on the 13th floor on Friday the 13th. You got these huge whipping winds coming through the lab, which probably wasn't so great.
And the occasional peregrine falcon.
Yes, right.
Yeah, we have windows that are locked. I think you can still do that, Alan. There's nothing different. And then once you unlock them, they swivel.
Right.
So they can clean them, ostensibly, but they're never cleaned.
They're never cleaned, yeah.
Right.
And that way. Yeah, I remember the lab windows being open that way. You have to be careful, though.
Oh, yeah.
Very
careful. You can lose stuff.
Yeah.
Like faculty.
Yeah.
Right, so it is warm here, but we'll...
Oh, and it's pouring rain here.
Oh, yeah?
Is it really?
Yeah.
We had that this morning, yeah.
We
It's
did.
66 Fahrenheit, 19 C, and the dew point is 64 Fahrenheit.
Oh, my.
So pretty much clouds are on the ground at this point.
Yeah. Well, that's the weather we had this morning.
All right, we have an all-email episode today.
Sounds like fun.
Dickson, did you say you did not want to read the Robin's email?
I'd be happy to.
Okay, why don't you? Because it has a reference to you in it.
Always. Something I must have said. Here we go. Robin writes, yet another multiple sclerosis treatment. A letter writer's sister has multiple sclerosis. Polyclonal vegetables should not be harmful and might even be beneficial. And then he gives several references to that. Minding your mitochondria in colon, Dr. Terry Walls at TEDx Iowa City. That should be an interesting listen and look. Mortality in biology. Remember, nucleotide-based life is geared to promote and perpetuate nucleotides. That makes sense. The fitness or otherwise of the genotype is based on the interactions of its associated phenotype with the milieu. The phenotype, in effect, being the interface. The cells, parens and virions, or virions as it's pronounced, organisms, social groups, nations, and empires are emergent phenomena associated with the phenotype. In spite of all their varied scales, they all feed back to the genotypes. Some of our DNA sequences go back as far as the last universal common ancestor. From the perspective of nucleotide chemistry, that's where the action is. All the rest is supporting and ancillary stuff, some of which is quite important at our scale. Natural phenomena may have a long reach at scales that are barely comprehensible to us. And for Dr. Dickson, the oldest living things in the world, photographer Rachel Sussman, interviewed by Ira Flatow. Science Friday. That's great. Oh, that's really terrific. So it gives a reference for that too, or she, depending on who Robin really is. And,
It's a guy. It's a man.
It's a guy. Okay, fine. I'll be on that like
white on rice.
I was going to say that, but it's such a cliche.
Like flies on
That's also a cliche, but as a parasitologist, I can appreciate that at least.
Right.
So this movie is a TEDx talk.
Right.
This is this Dr. Terry Walls. He talks about this woman with MS who, he says, obtained a cure by eating a hunter-gatherer diet, but in fact, it's not a cure. She just ameliorated some of her symptoms. Whether or not it had anything to do with her diet is absolutely not clear.
Right.
But I think Robin is, poking a little fun at it. Polyclonal vegetables, you get it? Because last time we talked about monoclonal antibodies.
Exactly.
Terry Walls is the physician in the video.
Right.
That's her name.
Right.
Is she the person who had
Yes.
MS?
Yes.
Yes. She's the MD in Iowa.
Got it.
Well, she didn't get cured because apparently she still has MS. But
Right, but it certainly seemed to have ameliorated her symptoms.
which is
But
good.
The problem with any relapsing remitting disease is that it relapses and remits.
That's right.
Yeah.
So anything can appear to be a cure if you happen to do it at the time the disease was remitting. I mean, it's nice that this worked, and it seems like probably a harmless intervention, if nothing else.
Sure.
It's fine to eat vegetables.
Polyclonal
Sure.
Yeah.
vegetables are great.
She also suggests seaweed and grass-fed meat, and the one I couldn't do would be organ. I mean,
Right. Right.
organ meats.
Yeah, I couldn't do that either.
But lots of people could.
Yeah,
Yeah,
liver
no,
and onions. Ugh.
No, I'm not a liver. Nope. All right. The next one is from Connor. Kathy, can you take that?
Sure. Dear Vincent, Rich, Alan, Kathy, Dickson, and guests, I just want to take this opportunity to thank you all for such a fun and informative blog and podcast. I, too, have experienced a TWiV bump of sorts recently, in that as of last week, I've passed my PhD Viva,
Nice.
and as of February, I am a postdoc at the MRC Center for Virus Research in Glasgow.
Fantastic.
While I cannot prove it, I am convinced that reading the blog, listening to the podcasts, and being allowed to be a part of 2 TWiVs shaped my personal and intellectual development as such to help me complete my PhD and pursue a research career in virology.
Wow.
As part of the first generation TWiV, I would like to thank you again, and long may it continue to inspire the generations of scientists to come.
Outstanding.
That's nice. Connor, of course, is Paul Dupre's student or former student. First generation TWiV. That's a possible title.
How wonderful.
I like
F1. Just F1.
Don't you think all virology graduate students now should have a TWiV experience?
Well,
Yeah. Yeah, it should
some
be
kind.
part of the program to be accredited as a microbiology or virology program.
Congratulations, Connor. Good work. Keep it up.
Yes, good job.
Alan, take the next couple.
Oh, yes, the next one is very short. Rehan writes, Hi, Twivers. This is a comic I found online. Just wanted to share. This is funny. You must check it out. To vaccinate or not to vaccinate? That is the incredibly stupid question. And this is a comic on a site called 9gag. A woman saying, "No, I don't want the vaccination for my baby. It causes autism." Doctor says, "But ma'am, this is a vaccination against autism." And the woman gets this panicked look, and ignorance intensifies. What are you going to do now? Yeah,
That's right.
It's good.
You might say he was needling her.
Oh, that was a shot in the dark. All right, John writes, I thought you might like this article on Slate. A brief, terrifying history of viruses escaping from labs. Did we talk about this one already?
I think so, yeah.
Or we talked about it on email? Did we talk about it on the show?
This is a version of the one in the bulletin for the atomic scientist. Yeah. This is a shorter version of this. Right.
Yeah.
This one appeared in
Yes.
Slate, right?
Yeah.
Yeah, this is all about the dangers of doing
Yes.
scientific research.
Scary it is to do research on viruses.
There is something here so my pick is related to this, actually.
Okay.
And we'll come back to it. But they talk about the medical photographer at the University of Birmingham in the UK, the last victim of smallpox. It was a lab infection, but we'll talk about that later. Yeah. All right. Do you want to do another one, Alan?
I'll do another one since those are both short. Robin again writes, how long can viruses survive? Crystallized viruses placed in a vacuum and protected from electromagnetic radiation should last indefinitely. But even eukaryotic cells can be deep frozen. However, viability is related to the ecosystem in which the entity lives. A human cubicle serf from a metropolitan area taken in a time machine back to a time and place before the first Ripidistia clambered ashore would be non-viable. The same would be true for most, if not nearly all, of today's tetrapoda. Any biological entity in nature evolves pari passu with its ecosystem. Isolated from its ecosystem, the mutual influences on shaping evolution cease. Even if the isolate is allowed to reproduce, its evolution will be shaped by the altered influences. If not allowed, its functionality will be frozen in time. A virus that is viable after prolonged isolation has dependencies for which the ecosystem has not changed sufficiently over that time. This could be related to the virus bringing along a lot of its own tools and equipment.
Right.
Okay.
This has to do with Siberian virus that we mentioned probably a while ago.
Right. So if the ecosystem in which that virus survived is gone, and you try and bring the virus back,
Right.
it's the place and time for which it is adapted no longer exists. So it may no longer matter whether that virus is reactivatable.
Right.
And then Robin goes on another topic. The 4 laws of thermodynamics are about energy. The 0th, connected systems will come into equilibrium. First law, it can't be destroyed. Second law, it turns to trash. Third law, it can't be cleaned up after it is trashed. It's a pretty good way to summarize them. However, matter in the presence of energy gradients is disturbed by energy flows. Some of the resulting random configurations may follow dynamic reconfigurations impelled by energy flows. The dynamic configurations that best enhance the dissipation of energy gradients are selected for, and even more so when those configurations can directly move matter into like configurations, hence chemical, biochemical, and biological evolution. Okay, so a thermodynamic explanation for how things evolve.
Right.
And then gives a website, Into the Cool, which is
cool. Energy flow with
about energy
thermodynamics
flow.
in life. Yeah, it's good.
Yeah.
Which, by the way, was the name of an album by Gil Evans at one point as well.
Really?
Oh,
Yeah, Into the Hot and Into the Cool. He had 2 of them.
It's a jazz guy?
Yep,
Are you a jazz aficionado?
I am.
he's from New Orleans. He has to be.
Is that genetic?
He was only there for 5 minutes.
You only have to be there for a millisecond and you're cooked. Too bad.
Continuing with Robin, talking about feeding on viruses, and he compares prokaryotic biomass and viral biomass. And prokaryotes are 2 orders of magnitude more than the viruses. The ecosystems must do something equivalent to eating to recycle that viral biomass. Okay.
Yeah, someone wrote about how there's so much virus out there. Does anyone eat it? I don't think anyone eats it, but certainly it's recycled, right?
Oh, wait. People eat baculoviruses when they eat cabbage.
That's true.
Yes.
Uh-huh.
Do we digest them? I suppose so, right?
Oh, I think, yeah.
Could you live on baculoviruses alone?
No, man cannot. No, I'm sorry. I can't degenderize that statement. But
humans.
People cannot live on viruses alone.
that's right.
They've got protein. They've got nucleic acids. You could do pretty well.
Right. I wonder what a plate of virus might look like if it was so much that you could visualize it.
A lot.
Yeah.
Well, all the viruses on the planet would fill Giant Stadium like 100 times.
What would it taste like?
I don't know.
Chicken.
Chicken.
Just
Chicken.
we tend to be wondering often.
Yeah, it's another title. I like that title. It doesn't matter, Dickson.
Tastes like chicken.
It tastes like chicken.
Tastes just like chicken.
All right. Robin is then talking about articles that are subject to reviews usually have a very high information density. It said if you copy from one source, it's plagiarism, 10 sources, it's a topical discussion, 100 sources, a subject synopsis, and 1,000 sources, a comprehensive literature review.
Very nice.
And then continuing the discussion of proximal and distal. Proximal, like distal, implies directionality, upstream or nearer to the center or source, more like a vector than a scalar quantity. In this case, it would refer to some point between the microphone itself and the sound system to which the microphone is connected. More proximate to the microphone would be nearer to the microphone.
Hey, Dickson, get nearer to the microphone.
I'm as close as I can get without being behind it.
That's what this is all about.
Yes.
Understood.
All right. The next one is from Anne. Dear TWiV team, wondering if you saw the article in the New York Times last week, which could be many weeks ago, watching proteins do the jitterbug. It was in April. The article included links to wonderful animations of cell contents and structure and protein packing. April 10th. It's a very nice article by Carl Zimmer. And she gives also a link to one of the embedded videos, The Inner Life of a Cell. A cool article about what animators are doing to give us an idea of what the inside of things look like cells. This sent me down the rabbit hole of YouTube videos on related topics, and I also found these videos. I did a quick search on the TWiV site to see if these had been submitted before, but didn't explicitly find these, so here you go, but feel free to omit. Flu attack, how a virus invades your body, and the deadly dengue virus video. And these are videos made by a company called Ex Vivo in collaboration with Harvard University. And I think we've mentioned some of them before. I've actually had an email from one of the scientists who collaborated on the dengue video. They're very cool. I use them in my lectures. So thanks for that, Anne.
Cool.
That brings us back to Dickson.
Okay. CN writes, greetings, professors. Multiple times has the discussion come to the topic of, quotes, what is not working in science? The funding, the publishing, and the way science is done. Often when I talk to my fellow researchers, I get mixed opinions. So I put up an online questionnaire to see what people have to tell. The sad part is the response has been very low. So I thought, if TWiV could give me some boost through announcement, that had to be a great way of finding out what others have to say. The link to the questionnaire is, and then that gives the link. With some preliminary polling, I wrote what I could gather, and then there's another link. If I could get sufficient response from all around, I want to put the results out there in the web, and sorry for the self-promotion. Thank you for all the free, super quality education on the microscopic life. It just keeps getting better and better and better. A Twix fan.
Yeah, so this is a poll everyone should go over and check out. It's basically about people who are doing research, questions about funding and teaching, research, how much time, this and that sort of thing. So check that out. That's not relevant to you, Dickson, because you don't submit grants anymore.
That's probably
But you could fill it out anyway.
I could do it.
I know you have spare time.
I have lots of spare time.
It's not self-promotion, CN. It's okay. And Matt Freeman, who had been on a couple of weeks ago, left a note here on this one. I've been wondering what we would really be studying if most labs weren't working on data for the next grant and instead working on the questions that really intrigued us.
Ha! Thank you, Matt.
It's really sad that we have to, we just can't pursue our interests. We have to, write things that are fundable.
Well, I wouldn't say they're not our interest, but I would say that
I agree. But
every now and then you get an idea that you can't work on because
that's right.
It's just not fundable.
That's the way to put it. You're right. I am interested in the things I'm funded for, but I'd like to be able to wander off and do anything.
Sure.
Okay. Yes.
Maybe not have to spend so much time writing the grants to get to do the work.
Yes.
Correct.
Yes, many people write every cycle 3 times a year, right?
Yeah.
That's
That's ridiculous.
That's crazy.
I mean, that's terrible.
You know what really touched my heart, so to speak, when I was on the study section, to see how fast 3 months of work goes down the drain.
Yeah.
You're sitting there, you read this grant, you read the review, the other reviewer gets up, they say ditto or they don't, and then they give their review, and then you sit down, and then you vote, and then you throw this grant over your left shoulder into a big giant box.
We don't have boxes anymore. It's all electronic.
I know, but they used to have boxes.
Yeah, I know, Dickson.
And you see this box fill up, and when it fills up, it's time to go home.
I know. After each round, everyone would fling the...
Exactly. They couldn't get rid of them fast enough.
And what I always hated was it was really early on before the days of recycling, people would throw their coffee cups in there.
Yes.
There's
Kathy,
coffee
you're right.
stains all over people's
You're
glands.
right. You're right.
Oh.
And that's somebody's
Very
life.
sad.
Those were some people's lives. Yeah.
Kathy, you're next.
Kevin writes, "Hello, Twivers. I'd like to chime in on the discussion from the last email episode 280 about the What is Life arc. Rich's pick of the week was an article saying that defining life is futile and unnecessary, and Vincent said he disagreed and that there actually was a clear definition of life and that it's useful to have. I think both the article and Vincent are partially right. The definition of life, like most of our definitions, is a model of reality. It doesn't describe reality itself. All definitions break down at the edges. Most textbook definitions of life would exclude viruses, but it's absurd to put viruses in the non-living category. Alternative definitions like Rich's from the same episode, that living things can convert energy into heat, would seem to include things like stars that we do not consider living. To take a less complicated example that illustrates the same point, try to come up with a clear definition of chair. What you'll find is that most definitions are too specific and exclude things that are legitimately chairs, or too broad and include things like tables.
Right.
I have a definition of a chair. It's what I spend 8 hours a day in.
That's right.
I guess it could be my bed, too.
The chair is the one thing.
No, I don't get 8 hours of sleep. Sorry.
However, this is not to say that trying to come up with a definition and arguing about it is not a useful exercise. We use definitions because they help us bring order to our fundamentally chaotic experience. Without definitions, everything we encountered would be a completely unique instance, and we'd never relate anything to anything else. However, like experimental models, the danger is when we forget that the model is a model and believe that it describes reality more accurately than it does. For the definition of life, perhaps this is somewhat pedantic, but I see it happening in my own field of immunology all the time. Cells are defined by certain cell surface proteins, and then someone finds a new subset that falls outside the definition, or falls inside only due to cell surface expression. And conclusions are drawn and rigidly held due to solely the original definition, which was made up in the first place.
Right.
Cheers,
I think
Kevin.
what he says is right. Our world is chaotic, and we define things to try and organize it. And that's why I think it's worth trying to define life.
Yeah.
Not that you're going to have a rigid answer, but it's worth the experience. And that in that Times article, I think it was, or opinion, he was saying it's not even worth talking about.
Right.
Which I don't think is true.
Actually, I would throw in my 2 cents here and say that life is definable by the fact that it has metabolism.
Dickson, is a red blood cell living?
Nice day out there, isn't it?
Why do you ignore me? I just want you to answer.
Well, we've raised that question recently. We did raise that recently on a TWiP. And, is a red cell, it depends on if it's nucleated or not. It depends on if it's expressing metabolism. All right? Metabolism is the name of the game, I think.
Evolution is part of it, too.
Metabolism.
Okay, I'm sorry I asked you.
I'm sure you are. But I gave you an answer anyway.
So I just want to get back to the last thing that Kevin said about immunology, where a certain cell type has a certain kind of marker until that kind of marker is found on another cell
That's
type,
great.
and
Right,
it just kind of blows the definition.
Sure.
So
I
people
like that
always
because
have
I've always had trouble with immunology because of that.
What's so funny is to see how many of the interleukins there are now compared to, let's say, 10 years ago when everybody had their own networks all made up based on the number known. And as soon as a new one comes in, it completely reorders the way in which the interleukins interact. And what are we up to now? In the 30s?
It's 22C right now.
No, I don't mean that. I don't mean that. You know what I mean.
Kevin had written to us before because he's one of the curators of the immunity.org site, and they send us a link for their immunity podcasts. It looks like they've done about one a month since then.
Cool.
So, yeah, they're sticking with it. So that's good.
Good.
Check them out.
That's excellent.
Immunity. EMMUNITI.
Unity.
On the other hand, my student's podcast is now defunct.
Oh, dear.
Biochemistry Revealed.
They have no more metabolism.
That's right.
So that's not life.
Alan, the next one is yours, and what could be better timing?
Yes, perfect. Michael writes, Esteemed doctors, I want to thank you for mentioning my microbiology history blog, Amboceptor, as a pick of the week a couple of months ago. And also thank Alan Dove for being one of the blog's first followers. They say that if you want people to start reading your website, the best way is to have started writing it 7 years ago. Nonetheless, it seems like I have a new Twitter follower every couple of days, and it's gratifying to be able to converse with established members of the science writing community. Although I'm trained as a virologist, it's often hard to write about virology from a historical point of view because our knowledge of the subject has developed so recently that even a paper from the 1950s is hard to understand. So unfortunately, the majority of the posts on Amboceptor would appeal more to TWiM than to TWiV. For example, he links to a few posts, one on how long does typhoid last in ice cream, which is quite good, the care and feeding of typhoid carriers, and Taiwao Bozu, the bald geisha plague of 1901. But this week's post, this is from a few weeks ago, Yellow Fever Stops at the Miami Airport, was particularly inspired by things I heard on TWiV, in particular Dickson's discussions of West Nile virus. Dickson may also be interested in a post from January in which I try to comprehend some experiments on polio survival in the housefly stomach from the University of Michigan in the 1940s. And that post is titled, Look at the Polio Fly.
That was actually for Vincent.
Yes.
Yes.
That's right. Dixon would be totally disinterested in that. No, actually, would be very interested in that.
Thanks for all your podcasts from a long-time listener.
The article is by Tom Francis, who did the the Salk vaccine trial at Michigan.
Wow.
Yeah, so, and it's a good blog post. What I like is that Mike tries to get into the data, and for this one, he analyzes a table and then just takes the data and puts it into a probably Prism graph or something, Prism software, and just gives you graphs about it. And he really tries to make it comprehensible.
Yeah, this is cool. He does a lot of this sort of thing. It's like literature archaeology. Goes back to the old days.
All right, the next one's from John, who writes, "Hey, Vinny and the capsids."
That could be an episode title.
I like that.
Vinny and the capsids.
I wanted to comment on the Mother Jones vaccination rate article from TWiV 278. The map of state vaccination laws is at best misleading. Compare Oregon and Massachusetts. The map says both are medium difficulty, requiring a note from your doctor if you don't want to vaccinate your kids. The doctor's notes in these states are very different. In Massachusetts, the doctor must say that vaccination would endanger your child's health. The medical exemption must be renewed every year. In Oregon, the doctor must say that you heard about the benefits of vaccines and still chose not to vaccinate your child. Before this year, you didn't need a doctor's note or counseling. Sounds like Oregon makes it easy to opt out. Now let me put the opposite spin on it. Alan's statement that Massachusetts does not allow religious exemptions from vaccination, TWiV 268 at 1 hour 34 minutes, is not correct. State law allows parents to claim a religious exemption without a doctor's advice. MGL 76-15 linked below. I guess that's a law, right?
Yes. Massachusetts general law.
If he lived in Oregon, he would have to listen to medical advice before claiming a religious exemption, SB 132, linked below. Now it sounds like Oregon makes it harder to opt out. Here's the reason for the difference. If a state allows a religious exemption, anybody can claim it. The state is not allowed to judge the merits of a religious objection. In Oregon, many parents claimed religious objections that were really medical objections in disguise. Vaccination rates fell low enough to cause a public health problem. In response, the state required parents to hear why their medical objections were unfounded. In Massachusetts, there is no public health problem. Vaccination rates are high, and state rules bar the few unvaccinated children from school during a disease outbreak, 105 CMR 220. Only 2 states do not allow religious exemptions, Mississippi and West Virginia. These are religious states, but no mainstream American religion opposes vaccination in general. Complicating this discussion is the existence of multiple overlapping vaccination programs. In Massachusetts, town boards of health can order mandatory vaccination if they provide free vaccine. There is no religious exemption in this law, only a medical exemption, and he cites the law. A more interesting story from Mother Jones is from 2013, explaining that the main reason for missed vaccination is much more mundane than religion or McCarthyism. Even in America, getting children, parents, doctors, vaccines, and money to the same place at the same time is a difficult logistical task.
Absolutely.
I'm actually writing up a meeting that covers quite a bit of depth on that very topic.
Yeah, it's difficult, but it's been done.
It has been done, but it's an ongoing problem to to get people into a place to get vaccinated in a timely manner, and especially with the reimbursement problems that have plagued healthcare in America. That is definitely an issue.
Yeah, but we do pretty well despite all that.
We do pretty well despite all that, and there's ample evidence that a lot of the problems that we've had lately with vaccination coverage, especially for pediatric vaccines, have been due to the misinformation campaigns.
Anyway, he provides links to all these laws and articles that he cited, and thank you, John, for a very comprehensive response.
Yes.
We appreciate the time that you took to put into that.
Yep.
Dickson, you are next.
Okie doke. Christopher writes, "Hey, TWiV team," or Vincent, whoever reads this. "So I got into a debate with my teacher today who told me interferon has no side effects. Obviously wrong. She also claims to use low-dose oral interferon for influenza prophylaxis, and I can only find one study, paid for by the pharma company who sells lozenges, close parens, that shows it isn't at all that great. Do you know of any studies that really discredit this peculiar therapy? I really don't see it working. Maybe you guys can give me insight on whether it works or not. Your faithful follower, Christopher. P.S. This was the article entitled Low-Dose Oral Interferon Alpha as Prophylaxis Against Viral Respiratory Illness, a double-blind parallel controlled trial during an influenza pandemic year. Versus...
No,
paper.
Stop.
Okay.
These are my notes, Dickson.
I'm sorry. Then I can read the next one?
No. You have to talk about this.
Fine. Talk.
The rest of this letter is my comments.
Talk about it then. Just go ahead.
So, interferon is a problem because it has side effects. It makes you feel lousy. Mm-hmm. Even when you get an illness and you make interferon, you feel lousy because of the interferon. So
lousy is kind of an understatement.
Yes, you feel very badly. So the hepatitis C treatment up until this time has involved interferon and very, very, very bad side effects.
It can actually cause deadly cases of depression.
Dear.
So to use it to treat influenza would not be a good idea. Nevertheless, there have been a series of trials, first in animals. There have been human clinical trials using interferon either intranasally or injected, and they show some benefit. All right. So it's not without merit. You can't discredit the use of interferon. However, since we do have antivirals and we have vaccines, and there are new antivirals being developed, I would doubt if interferon is licensed for this use.
So does interferon itself make you feel bad, or does interferon's induction of other things?
Yeah, the things it induces, for
like
sure.
TNF.
Well, it induces over 1,000 interferon-stimulated genes,
exactly.
which, Dickson, we work on in my laboratory.
Yeah, no, that's great.
I just wanted you to know.
I think that's fantastic.
And those have side effects, yes, for sure. And the complicating this is that not every strain of influenza is susceptible to the same levels and the same types of interferons, et cetera, et cetera. Yep. So I don't think it's a good therapy because the interferon response is probably tailored by us to fit the pathogen. And if you're just dumping interferon,
it's not targeted.
People,
it's generic, and that's what's wrong.
Exactly. So,
and I don't know what these low dose lozenges that this person is taking, what the dose is, and how much the effective dose is actually getting into the bloodstream.
Exactly.
This sounds like kind of a load to me. But as Vincent said, this is not a priori discreditable.
Well, Kevin, you got a problem with that teacher.
There is a drug. Let's see, that he says is I found a drug called Gripferon, which is a nasal drop.
Good name.
Gripferon.
Yes.
It's manufactured by, I can't tell the company, I don't know. No. Anyway, you can apparently get this somewhere in the world. And if you look at, there's a Wikipedia page which lists all the interferon products that are either being tested or approved for various uses by different companies. So.
Can I go back and read the word lozenge because I didn't read it correctly last time? Could you please put that in place of the word that I tried to pronounce, thinking it was the name of a generic drug?
Dickson, what are you thinking?
Lozenges.
Lozenges. Okay, that's fine.
Have you had your lozenge?
Kathy, you're next.
Okay, this is Fernando. Fernando writes, "Hi, Vince and friends. Your occasional comments about tighter research funding and decreasing support for fundamental science agree with my observations of my own field, computer science, as well as with my experience during my academic foray into computational biology before moving back to industry over 6 years ago. I just came across a recent PNAS article, and he gives a link to the Alberts, Kirshner, Tillman, and Varmus article. He says, I think it describes the situation well and makes some interesting recommendations that match some of what you've said in the podcast. I feel you could have written this quote. Overvaluing translational research is detracting from an equivalent appreciation of fundamental research of broad applicability without obvious connections to medicine. Many surprising discoveries, powerful research tools, and important medical benefits have arisen from efforts to decipher complex biological phenomena in model organisms. In a climate that discourages such work by emphasizing short-term goals, scientific progress will inevitably be slowed and revolutionary findings will be deferred. Please consider this as a listener pick. And in fact, we discussed this on TWiV 282, and I know that in my copy of the article, I have that paragraph underlined, and I can't remember whether I brought it up, but we were also asked by the current president of ASV to comment on the article because she was going to be in a conference call with presidents of other societies, and she wanted input from us, and I highlighted that comment as being a good one.
I sent her a link to TWiV 282. I said, basically, we concluded that nothing will happen without leadership from the top of NIH.
Here.
Okay, thank you, Fernando. Alan, you're next.
Okay, this is a long one. All right. Longtime listener, first-time writer, Lisa writes, in episode 278, Andrew wrote in about island biogeography with ideas about how it could relate to viral ecology. I just love that people are thinking about these kinds of ideas. I'd like to take a moment to help flesh out some of the background of the ideas you were discussing. Andrew hypothesized that larger animals may host a greater number of viral species than smaller animals. Alan responded there are a limited number of niches in an organism. Presumably there are similar numbers of tissue types in mammals ranging from mice to elephants, so despite the vast difference in biomass, the number of niches remains roughly constant. However, in organisms with vastly different physiology, the number of niches may vary greatly, such as a sponge versus a mouse. Alan's response harkens back to Gauss's classic experiments with paramecium, in which he demonstrated that the number of species or phenotypes with different resource use that could coexist within an environment is limited to the number of resources, i.e. niches, available in that environment. If there is only one resource, then the fitter of the two species will competitively exclude the other. There are also examples of cross-feeding where two phenotypes can coexist on one resource. One phenotype uses the single provided resource and the other uses the waste from the first phenotype. This competitive exclusion principle holds true at equilibrium in a well-mixed environment, that is, when the environment is constant. In nature, Darwin's finches appear to follow this rule, but by and large it is difficult to find other cases so tidy. The classic example of this is the paradox of the plankton. A variety of species of plankton can coexist within a community on a limited set of shared resources, nutrients from seawater and sunlight. Several explanations have been put forth to account for the coexistence among competitors in nature, such as niche partitioning, spatial heterogeneity, and disturbance. Bottom line, nature is complicated and does not necessarily follow the rules we observe in the controlled laboratory environments, but that does not mean that we cannot glean important knowledge from lab experiments, which allow us to isolate specific mechanisms and test them directly. So back to Andrew's question. Yes, the number of viral species is limited to the number of niches in theory, but in practice, I think we cannot simply assume that just because mice and elephants have the same number of tissue niches, that they also harbor the same diversity of viruses. Perhaps the larger size of an elephant will allow for greater spatial heterogeneity, for instance. The jury is still out. I'd be curious to see some cross-species comparisons of microbiome diversity in general. Bacteria will also face limitation in the number of niches, but I'm not as familiar with that field. Vincent also gave a great example of a retrovirus using a surface protein to inhibit other viruses from attaching. This is a perfect case of interference competition, where one species directly prevents its competitor from accessing a shared resource. Another form of competition that viruses could experience is exploitation competition, which occurs when a virus, or anything else for that matter, exhausts a shared resource before its competitor. Vincent mentioned somewhat dismissively that a few studies have been done in the lab. A few studies have been done in the lab. I'm working on one such study for my dissertation work. And this explains why she's so well informed on this topic.
Indeed.
I'm currently a fifth-year graduate student, almost done, using an experimental evolution approach in bacteriophage to study competition as a key driver of diversification, and specifically the evolution of novel resource use.
Nice.
When viruses evolve to utilize a novel resource, they're evolving to infect a new species. We found that novel host use is more likely to be selected for when there is competition for resources present, and gives a citation. We've also been looking at how the strength of competition affects the ability of multiple phenotypes to coexist and how gene flow affects adaptation to a novel host. I suspect that by the time I defend, I will have counted hundreds of thousands of plaques. I've been looking for work on how my work relates to viruses in nature. I happened to hear a relevant talk at the Jacques Monod conference series entitled From Emerging to Pandemic Viruses, the Interplay of Host Ecology and Viral Evolution, which you advertised on this very podcast. Great meeting, by the way. Gail Thibault presented work from his group, gives a citation, that examined the competition between two viruses in nature. They observed that when a second viral strain invaded an island, both the old and new strains could coexist. This presented a conundrum. Why didn't the new, fitter strain win out? They found that during co-infections, the old strain actually has a higher fitness. This allowed both the old and new strains to coexist. Pretty cool, huh? Thank you for all your hard work on these podcasts. They've really helped expand my background and understanding in virology and microbiology and make the hours of pipetting go by. The weather here is a perfect 74 degrees Fahrenheit, 23 degrees Celsius, under blue skies, but very high pollen here in beautiful Chapel Hill, North Carolina.
Right.
Cool.
So I think I was dismissive because it's hard to study these things in nature, but she makes the good case that you can learn things from laboratory experiments, and that's in fact what she did in her work, and which is really interesting. It shows that when you subject a phage to competition, it evolves to infect a different host. That's pretty cool.
It's neat.
Very nice stuff.
The phage she works on is called phage 6, or just 6 for short.
Yeah.
I've never heard of that one before. It's cool.
Yep. Okay. And she says she gives us the literature citations for these. It's interesting, that meeting, the guy had asked us to advertise and she went to it.
Nice.
Cool. This is all a big web, right, Dickson?
Indeed.
I meant to ask you, Dickson,
Yes.
so in parasites, do we know whether large animals have more parasites than different parasites, different numbers of different parasites than smaller animals?
We don't. We don't know the exact answer to that question, but I can tell you that the larger the animal, the larger the tapeworm.
Okay.
So
Okay.
the size of the parasite
Yeah,
varies.
that's fine,
with the
but I'm
size of the
thinking
host.
of whether there are
I know
different
what you meant.
niches and that you could have different kinds of parasites.
No, but certain groups of animals have very few parasites. They are very clean and neat eaters. For instance, crows. You'd think crows would have tons of parasites. They pick out the contents of the stomach of the killed hosts along the side of the road. They're eating dead insects. They're eating a lot of things that don't give them parasites, but they also get a lot of viral diseases and protozoal diseases that are transmitted by vectors. So some groups are more susceptible to vector-borne diseases, others to eating disorders because of what you've just ingested.
Right, so the answer is you don't know.
The answer is I don't know.
Okay, thank you.
But I love taking up time just saying I don't know.
No, I think it's a similar question with viruses and eukaryotic parasites.
But it depends on the roots and the kinds of exposures and that sort of thing. So, I mean, the dirtier the environment, the more parasites you're going to have. The cleaner the environment, the less parasites you're going to have.
So it's a difficult
It is
correlation
difficult.
to
Like
make,
human
right?
beings are, one species supposedly, but if you look around the world,
But if you look on the plains of Africa
that's
at
right, that's
an
right.
elephant versus a mouse,
yes,
does the elephant have more parasites, diversity than the mouse? That's the question. It hasn't been done, I guess,
don't ask the elephant because it just ran away.
from the mouse. Yeah.
Mouse, yes.
Interesting that I would pick those two.
Yeah, that's exactly right.
Next one is from Kurt. Hello, TWiV team. I would like to begin with a thank you to Vincent as the Virology 2 course comes to a close on Coursera. The experience was as entertaining as it was educational.
Oh, nice.
Wow. I'm hopeful we'll see even more microbiology on there soon. I was recently pondering the inability to grow human norovirus in cell culture that you mentioned in the course, and well, I hope this train of thought makes sense. Caliciviruses seem to be primarily at home in aquatic environments, but what keeps the virions from scattering hopelessly in environments such as the open ocean with whales? Worse yet, according to an article in Applied Environmental Microbiology, murine norovirus is inactivated by room temperature water pressures of about 400 MPa, readily achieved at less than a meter in seawater in roughly 5 minutes. What's a virus to do? I'm operating on the assumption that formed virions take a small portion of cytoplasm when they form. Consider that noro and other caliciviruses may benefit from producing virulence by replicating in cells with reduced electrolyte loads secondary to diarrheal disease. The lowered tonicity plasma/cytoplasm in the virion may represent a lower density than the surrounding seawater, allowing the virion to stay near the surface where it's more apt to endure and get picked up by an unsuspecting mammal. To elaborate, pure water is about 1,000 kilograms per cubic meter. Seawater is 1025 at the surface, and typical plasma is also 1025 from what I've read. The more virulent strains would create more of an electrolyte imbalance. Presuming all electrolytes are shed with near equality, this leaves our virion with a more water-like, less dense cytoplasm. Perhaps this also lends a structural stability to the virion, which may help explain why the virus fails in cell culture, which requires an isotonic setting for the cells to persist. Hope that wasn't too silly. Thanks for your time and effort in doing this show, guys and gal. Pondering the microcosms with you helps the late-night driving. If appreciation could be cashed in, you would want for nothing. Thank you.
That's amazing.
Now, the neuro, I can't say for all viruses, but these caliciviruses are icosahedral, and I'm sure that the interior is packed with viral RNA, and there isn't probably much room for anything else, so I wouldn't say that it's capturing cytoplasm.
Well, I have another thought, too, and that is, what about Brownian motion? At that level...
It probably keeps them near the surface, right?
Well, it keeps them from settling. Do you think there's a rain of viruses in the ocean?
No, they're not heavy enough, no.
Exactly
not the small ones like this.
Yeah, that's exactly right. So I think that, electrostatic forces will keep them in suspension, and plus wave action.
I think that the inability to get this virus to replicate is just something to do with the cells that are being looked at. So
This thing with the pressure inactivation of the virus
that's interesting.
strikes me as really bizarre.
It is interesting, right?
Yeah.
It had never occurred to me that you could crush viruses.
Oh yeah, we saw images at the International Adenovirus meeting a couple years ago of atomic force microscopy and seeing this probe come down and touching the virus and then it just exploding. So this is a different thing.
So these folks, they expose the virus to these pressures, which I guess don't seem that extreme.
But if you're a virus, maybe they are.
And they got huge reductions in plaque forming units. If this can't, I can't think that this would be a general phenomenon. Otherwise, scuba divers would never get sick, right?
At least not from viruses.
At least, and not right after diving.
Right, but
Exactly.
you could cure all sorts of infections just by going down to 30 feet and looking at a reef for a few minutes.
Wait a minute.
It's a good question whether other viruses are similarly inactivated.
Yeah,
And I don't know.
and whether this would work in vivo.
It says here that they're using high-pressure processing for food.
Sure.
Right. They put the mouse norovirus into oysters, and so they talk about this could be a way to inactivate human norovirus strains in food.
So it's not enough to crush the oyster,
No,
because we don't want crushed oysters, right?
no.
No, of course not.
I'm looking here, but they don't talk about other viruses. So that's an interesting question. Whether polio, for example, would be similarly inactivated. How do you apply pressure? What do you do? How do you do it to a certain amount?
They have
wait,
ways.
no, this is not this pressure analogy in this letter is not correct. You would not get 400 megapascals in a meter of seawater.
No.
I just did a megapascal to atmospheres conversion. A megapascal is 10 atmospheres. or 9.8 atmospheres.
At what depth does it achieve that?
You get 9.8 atmospheres at 100 meters.
Okay,
So that's significantly deeper than scuba diving depths, and that's 1 megapascal.
sure.
So this 350, 450 megapascal thing is a high pressure.
Maybe they gave the wrong units. Instead of mega, they meant mini.
Or milla.
Well, in the paper, it's 400
Exactly.
capital MPa.
Right.
Yeah, I take that as megapascals.
Yeah, that's
Right.
correct.
They go to a lot of description in the thing about how they ship their samples with blue ice from their place to somewhere else, to Virginia Tech, and then they pressurize them in what sounds like just a pressure cooker or something, a Quintus 35L food press.
Huh.
Now you can get huge pressures in
You can use a French
fairly
press.
easily in industrial processes.
Well, a French press will definitely let you do that.
Right.
That's how they used to rupture cells.
I'm looking for this QFP 35L food
Yeah.
press. I guess that's the whole point. You put the food in it. And,
Right.
oh, look, here's one where they are. Actually, I did a Google and I got a proposal from Virginia Tech on the effects of hydrostatic pressure on toxoplasma
How about that?
Aha.
in ground pork.
Look at that.
How about that?
That'd be nice to try to see if that works.
Interesting.
Sure.
Okay, this is a new discovery. I want to do this for polio, but I can't justify it because there's no. I mean, in the intestine, there's not a lot of pressure. So,
Well, hopefully not 400
not.
megapascals.
No.
although sometimes Dickson there is, right?
Oh, sometimes.
All right. Did I read that? No. Who read that?
I read that. I
That's
think,
right.
didn't I?
You did?
Yes.
Really? That means I'm next?
Right.
Wow. Okay, Stephen writes, here's an interesting story. Cast a bearded dragon, a cricket colony, an iridovirus, an insect behavioral biologist, and Donald B. Stoltz, who works on viruses and parasitoids.
Goodness.
Parasitoid wasps. So this is an interesting story.
Oh, wait, I'm sorry. You did read the one from Kurt.
Oh,
Okay, continue. Continue. Sorry.
one from Kurt?
Yep, the previous one about the pressure. You
All right.
did read that one.
Okay, so I
But
read
anyway,
two.
you just
It's
read
fine.
one.
This is pretty short. There's a paper that was covered extensively in the press. It's called A Viral Aphrodisiac in the Cricket Gyrillus texensis. It was published in the Journal of Experimental Biology. Basically, it's an iridovirus of insects. It makes the crickets sterile, but they still mate. They actually mate more, and they think this is a way of allowing the virus to spread. What do you think of that, Dickson?
Cool.
Behavior modification.
Absolutely.
Males and females continued to mate when infected. In fact, infected males were quicker to court females than uninfected controls. Behavior modification, another example, right?
Absolutely.
Yeah, this was
Cuts across the entire
It's an interesting
phylogenetic
story.
tree of life.
And if you could adapt this to insects you want to eradicate,
Yep.
you might be able to use it for the sterile insect technique.
That's correct.
Yeah, figuring out how that works would be interesting. I presume it has to do with replication in ovaries and testes, but we don't know.
We don't know.
All right, Dickson, you are next.
Okie doke.
This is a very yes, Kathy.
Could I do this one?
Yeah, I was going to say. I was going to defer to Kathy anyway.
Yeah, I don't blame you.
Because I know this
Kathy.
person.
Oh, okay.
Please do.
Yeah. Nella writes, hi, everybody. You're listening to Nella from Hamburg, Germany, and what she thinks was very interesting about Vincent's interview. I am a student of infection biology at the University of Lübeck in Germany and spent 4 months in Ann Arbor, Michigan to work there, which was not that long ago. Maybe Kathy remembers me. Of course I remember her.
you
We
work
had
in your
joint
lab?
lab meetings, and she was here through January.
She didn't work in your lab, though.
No, she worked in another lab. And I think she came from a connection through Stefan, actually, the University of Lübeck. Yeah. I really enjoy listening to the podcast because besides all the really important and interesting information about virology, which I think is awesome, I also get to hear you guys talk about less important stuff like the weather and interact with each other. In my opinion, people who I met in the U.S. have an especially friendly, funny, positive, and open-minded way of talking to each other, and I am missing that a little bit since I am back home. Anyway, What I actually want to do with this email is comment on Vincent's dreams/plans for the future. If you should ever go through with the vision of building a company that tries to interact with the society and communicate science, please let me know. I am very interested in that, and I know from experience how important it is. Because my mother always taught me how bad vaccination is, which of course is not true. But that was the way she thought without ever really explaining it to me. Unfortunately, as a child, you think everything is true that your parents tell you. Now I'm 27 years old, and of course vaccinated, because I learned that parents are not always right. Even so, I love my mom. But Germany is in general a country where the rate of people who get vaccinated is going down, because the politics had some problems to communicate well with the public during the problems with the swine flu in 2009. At that time, there were different vaccines available for the public and for the people who worked for the government, and nobody understood why. Everybody thought that the government employees got the good and pure vaccines and the public got the cheap vaccines, which wasn't true, but nobody explained that to the public. So now a lot of people in Germany mistrust vaccinations. I think we should really learn from that and communicate better in the future. And there is so much other science stuff out there that is fascinating and great and mind-blowing, but people who don't work in the field have trouble understanding it and what consequences it might have for them, because we use all these complicated terms and talk to each other in a language that is difficult to understand for people who have nothing to do with science. I think that's really sad. So in 2018 or 2019, when I have my PhD in virology, which I am sure of, I would love to work for you, Vincent, if you are really going forward with your dream, which I think is awesome and a really good contribution to society. Even so, I know this email is a long shot because you might not realize your dream, and even if you do, you probably won't remember me, but that's okay. I will keep track of your career, and I will write you again if it happens. I also volunteer in a science association that tries to communicate science to the public. But as you know, you often only reach the people who are already interested in science anyway. It is still a lot of fun, and I always get reminded how difficult it is to explain science simply because the systems always get more complex. But I like the challenge. I hope to hear from Vincent someday. Have a nice day, everyone. Nela.
That was a nice letter.
Cool.
Very nice. Thank you.
Yeah.
Lovely.
Keep you in mind.
Right.
I am trying to do this, right, Dickson?
You bet. Okay, it's my turn now. James writes, Dear TWiV team, okay, I have written far too much recently. Parens, I type TW and you pop up in my address field, but here goes. One last time for a while, I promise. On TWiV 282, you mentioned the large intake of PhD students and job availability. Vincent, in particular, spoke about staff scientists. I left the biological field after graduation to work in the most tenuous, farthest fringe of science. I have two key points in relation to the discussion, one reflective of my experience, one of my current workplace. Note, some of this will be from a UK or European perspective, but I suspect not totally irrelevant. Number one, the number of postdoc positions not being sufficient is easily seen in my work. Until recently, last decade or so, those doing my job and directly under me would be high school leavers from the UK. Now they are invariably those with masters, PhDs, even multiples thereof, despite low pay and zero progression in most cases. For those working on the scale below me, almost invariably significantly less than 119 19,000 pounds. 19,000 pounds. That's UK pounds. What I have also experienced upon setting shortlisting tasks and questions for these is the quality. While this partly is a reflection of the salary, it isn't in all cases. A few are excellent. I have seen people who have gained scientific masters and PhDs from numerous countries, yet they have not attained the most basic of laboratory skills, possessing biochemical science higher qualifications, but are simply unable to calculate or make a simple solution at all. Never mind accurately to do this in practice, with no more success focusing in a simple slide on a light microscope. This brings me to wonder, just how did they attain their PhD position? Friends, I know there is variability between institutions, but come on. These are postdocs and skills I gained across the scientific disciplines before I even started my undergraduate biology degree. Number two, The proposed staff scientist position sounds like a perfect idea. Having always been commended for my ability, particularly in cross-linking with other disciplines and with a meticulous, quick, accurate ability to pick up practical science procedures to the highest of standards, and in considering the data. The fact that I have previously built high-end scientific equipment additionally helps me with my understanding on the technical level. But not being in a position to take the short contracts, I wouldn't like eviction. That would be my main pathway into science, and less than enough experience for direct PhD entry and unable to afford gaining a master's first or do expensive volunteer work. Alas, it is my fate. I'm slowly learning to live with it, although those in my work questioning, why am I doing only this job, doesn't help. The compromise of giving up a potential track and career progression would be counterbalanced by a secure income and more beneficial to working in a completely unchallenging environment for those in various similar situations. The staff scientist can also do something unique, concentrate on lab work. Not on the next grant, not on the next position, not on the looming end of contract. So the researchers knows the day to invest.
The day-to-day.
Thank you, Alan. Knows the day-to-day bench work is going on safe and uncompromised. In addition to the continually provided, you see, I'm having problems here,
continuity.
Folks. In addition to the continuity provided to researchers and the department, the finance invested by the university would not be lost staff. Scientists would be less likely to take their knowledge elsewhere. They amass more instead, becoming a pool of information to draw upon. A early career scientist is encouraged to move to get the next position at the end of the contract, and all the work involved in doing so in the last few months. A staff scientist would have less impetus to do so. Summary. I would concur on reducing staff scientists and or reducing the PhD positions. But does this experience not question the methods by which those are selected in the first place? From your valuable perspective, knowledge and hearsay has there become an initial layer of natural selection rather than the best skilled for the positions? A staff scientist would be a wealth of knowledge for a department, with no general factor to leave one institution to go to another, in contrast to an early career scientist. That, of course, in addition to the number of postdoc positions compared to those who completed a PhD. I wonder if valuable scientists are lost as a result in favor of those who can't ride the storm. I am working on being concise in the future. Thank you. I'll work on my reading skills also. Thanks for Twix, as always.
You know, this is something I've wondered about, because right now we impose a lot of selection, and you start out with X number of PhD students, but they don't always go to the end.
No,
and if
don't.
you just artificially reduce the number, what will be the impact?
Right.
I don't know. It's a good question. What do you think, Kathy?
Well, it's something I commented on yesterday when I wrote to Paula that, I think if there's going to be a truncation of the number of incoming PhD students, they're going to have to evaluate it at a lot of steps, because there are a lot of things that PhDs do besides research at R1 institutions.
I'll say.
And so I would agree that, maybe we have a kind of a glut right now, but we might not always. And if we make too drastic a cuts, then it could be a problem.
I wouldn't be too concerned about that. If there are jobs, people will come to fill them. And if you need PhDs, if you need more PhDs in the future, you can add more positions for them, and people will come in if there are desirable posts for them afterwards.
But what if they haven't been trained? I mean, we hear about there being physician shortages in the not too distant future.
Yeah, that's always in the not too distant future. You notice it never seems to come to pass. Now, that's not to say that there aren't localized shortages. There are parts of the country that are very poorly served medically, but then there are other parts of the country that are grossly overserved medically.
Sure.
New York City has been closing hospitals left and right because they have too many beds and they can't fill them. But you go to rural, parts of Mississippi, and you can't find a doctor anywhere within 100 miles. So it's a distribution problem. It's not a supply problem, and creating more doctors isn't going to fix it. It's the same thing with PhDs. You've got not enough jobs for the ones you have. There is, I believe, an oversupply. And yes, there are a lot of PhDs going on and doing other things, and I hope we're doing a reasonably good job at those other things. But was the PhD necessary for all those folks? If it was, then you can adjust later. But I think it is fair to say at this point that we are probably producing too many research emphasis PhDs. I don't know. That's just my assessment of it as somebody who came through the program and then ended up not working in a lab.
No, that's right. So I'll just comment briefly, because a lot of people that get their PhD can't find jobs in academia at the levels that they expect to find a job at. And namely, they want to be a professor, they want to have their own grants, they want to do a little teaching,
right,
and a little bit of academic administration, but mostly they just want to be paid to think. So a lot of those people, in addition to becoming what you became, Alan, they become high school biology teachers or high school chemistry teachers or high school. And those are real assets to those schools, because look what they know, and look what they can bring to those schools, a new level of knowledge that wasn't there before, that inspires a whole new generation of kids to go on to do even better than they could have done without those people doing that job.
But
so,
what about their PhD training actually prepared them for that?
No, it didn't. Of course it didn't. But
I disagree. I think that when you graduate with a bachelor's degree in science, you probably don't have the same critical thinking skills that you have when you graduate with a PhD.
I would agree with that.
And so I think having those critical thinking skills probably does raise the level of education in those schools where there's a PhD in a high school setting.
It has to.
One of my, in fact, my first technician told me, I don't know, about 5 or 8 years ago, that her husband feels like he was no longer really doing science because he's teaching high school. And I wrote back and said, au contraire, he's doing some of the most important science.
Yeah, I would agree, because you can at least distinguish the difference between an hypothesis, right, and a belief system.
But
that's very important to know.
I think you can improve teacher training without... I mean, the PhD does accomplish that, but it does it in a very roundabout way. I think you could accomplish a similar result more simply if that was the goal. And I don't see a big downside in reducing the number of PhDs we're producing. Of course, I say that as somebody who already got his. So take this all with a grain of salt. Yeah. It's like last one in closes the door. But but I do think that this addiction to graduate student and postdoc labor is something that's part of the problem now in the scientific labor market.
I agree, Dickson, that a PhD could make an effective high school teacher. Mm-hmm. However, I don't know if getting a PhD is something you really need to do that. I agree that it gives you a totally different perspective, but that's 5-6 years of research, which is really geared to do something else. If we wanted, as Alan said, we should develop different programs to make high school teachers better in the sciences.
So let me give you 2 examples that I know directly where it did make a huge difference, and that is the Bronx High School of Science. If you go over there, almost 80% of the faculty have PhDs.
If you go
to the Bergen County academies, they're called the Bergen Academies, over 60% of those instructors have PhDs, but they also conduct research.
Let's make a program that is geared to making high school science teacher, which combines, training to do that and some research.
Well, they're going to demand their PhDs as a result.
Well, that's fine. You can get a PhD. It's more advanced than other things, but it's not geared to making an R1 PI out of you.
No, that's correct. So they teach high school students how to conduct basic research.
But right now, here in this department, we're training people to be PIs.
Right.
And that's not fair if they want to do science, science teaching.
Yeah, you're quite right. And plus, the thing I find wrong about this program here, as opposed to the one I went through at the University of Notre Dame, was that I was obliged, before I actually got my PhD, to teach. I actually had to interact with the undergraduates. I don't see that happening here at all. And
Well,
you don't
we
learn
don't-
any teaching
We don't
skills.
have any undergraduates here.
No, but you do so. You've got a whole campus full. But we
As I said, we have no undergraduates here. They are
We-
at a different campus.
we-
This is a
Different
medical
campus.
school.
we don't
Dickson.
use our labor force the way we could.
Right, because you have to use it in the lab in order to get your grants renewed.
Yeah, exactly.
That's a
And
good
that's
point,
the
Alan.
essence
That's a
of-
good point.
I think.
Well, that's
I
one of
think
the points
a
of
big
this
part
paper.
Do you think
problem-
that's an indentured servant?
Kind of, and I think it wouldn't really be fair to saddle the Research One programs with changing their training program to produce more high school teachers, for example, or more science journalists, or whatever it is we think people ought to be doing with these degrees.
Sure.
Because you do need people working in the lab doing the work,
You do.
but having that structured as an academic training program to make more researchers is the mistake. That's what I'm saying.
Yeah.
It should not be, and that's the way those programs are structured. I mean, coming through Vincent's lab, I was taught, I think, in an absolutely world-class manner, how to be a scientific researcher. And they're just,
And now look at you.
and exactly, now look at me. They're just, if you keep training people to be the next Vincent Racaniello, where there's only one of him, and yeah, he will eventually retire, but then the 300 people who you've trained to replace him are going to be cutting each other's throats to get the job.
That's
And
today.
that's what we have now.
No, I know that. That's what we have today.
That's why the
That's true.
staff scientist is suggested, because
Exactly.
that
Sure.
does the work in the lab, and you
Yes,
don't need to
that's
have
right.
students to do that.
Yeah.
Have a separate career track for that.
That's true. But at least at the school I got my degree at, they considered teaching as an essential ingredient in becoming a research scientist. Because you're going to teach the postdocs. You're going to teach the graduate students.
I agree with that,
Sure.
Dickson. Totally.
So you should
Totally
learn how
agree.
to teach before you have to.
It's one of the shortcomings of this medical center
Yeah,
that
that's right.
teaching is not something that is
That's-
encouraged or rewarded in any way.
Exactly right. Exactly right.
So, some people have trained a disproportionate number of people to go on to do R1 research, and many of those people who are, some of the top scientists who've trained a disproportionate number, all of their trainees are employed like that. I've had maybe a dozen PhD students. One is at an R1 institution, one is in industry, several are teaching at small liberal arts colleges, and so, I'm not saying that everybody has to be like me, but I suppose there's to balance out the labs where they've produced 35 PhDs at R1 institutions over the years, there's got to be something at the other extreme,
Right.
or not, or there's got to be people doing research that aren't training that many students, which comes back to the idea of the staff scientists.
Nice. Okay. This was a good paper. It was a good discussion we had before, and this too.
Yes.
Alan, you're next.
Okay. Let's see. So that
Joseph.
was. Now I'm up to Joseph. Okay. Esteemed TWiV panel, I was pleased to hear you read a letter from my old lab mate and good friend Nathan on TWiV 282. I have very fond memories of Jacob Bernowski's excellent show from my childhood and was recently reminded of it by this interesting commentary in the New York Times. Just bringing that up, which contains an embedded video of the last few minutes of the Bronowski series. And this is, I have not read this. I have not read this opinion page yet. What is?
It's a guy who's reminiscing about growing up with Bronowski in the UK.
Okay.
Yeah.
Okay, intellectual modesty and self-criticism are indeed essential features of good science that can benefit wider fields of human endeavor.
Here, here.
Thanks again for the excellent work, Joe. P.S. It's a lovely Sunday afternoon here in New York. Who cares about the temperature? The dew point is a delightful 1 degree C, 34 Fahrenheit.
Yeah, it's a very nice reminiscence of what he learned from Bronowski. It's kind of nice. The dew point, huh?
Right.
That's a good measure, so it's nice and dry.
Okay, the next one is from Jacob, who writes, "Hi all, I just wanted to add my 2 cents to the negative results thing. I'm currently listening to TWiV 279, in which there were several listener emails about this while doing DNA extractions using a procedure that I developed that would not have been possible without other people publishing things that didn't work. When publishing a method development paper, people include a lot of information about things that didn't work because they compare several different procedures for doing something, and one or more of the options are more successful than others. The results for all of the options are published alongside the ones that did work. Even in a normal paper, there are a lot of results for things that didn't work if you read the methods and results. They just make the story of the actual results more interesting. I must admit at this point that I usually read the methods section of a paper first after the abstract to see if their methods and controls look legit before I read the rest. Sadly, some papers don't make it through. More broadly, if you use appropriate positive and negative controls, which you should be doing anyway, it should be possible to differentiate between the procedure failing and the result being negative. If you can't do this, you probably need to redesign your experiment. On the topic of having a database of things that didn't work, this might be problematic, seeing as though we don't even have a database of things that did work at this stage. It's great.
Good point.
On the discussion from a listener comment in this episode about successful vaccines preventing replication, I think that after listening to the Salk interview yesterday, there would be a good argument for saying that OPV is unsuccessful in that it cannot eradicate the disease, but it is successful in preventing polio replication in the gut, where IPV is not. The question is not if they are successful, but if they are fit for purpose for what they are needed for. On the general bashing support of peer review is not designed to ensure that your results are correct. It is meant to ensure that the conclusions you have come to are consistent with the data presented. Nice. As an aside, does Vincent pay attention to things shared to him on social media for TWiV pics and comments? I shared something with some exposition on either Facebook or Google+. I can't remember which, quite a while back, but I haven't heard of it on audio. Currently in Sydney, Australia, it's 18.6 C and cloudy. Jacob is a molecular biologist. I have tried to pay attention to suggestions on social media. I might have missed yours, or we might have, I don't know. I'm sorry, Jacob, but I'll try and be better.
Right.
I do try, and a lot of the suggestions I pick up on Facebook or TWiV or Google+. But not everything we do that's suggested, I must say, for various reasons that are not needed to go into at this time. Mm-hmm. Okay, let's do another round of letters.
Okey-doke.
Get back to you, Dickson.
Duncan writes, "Hi guys. I don't know if this article by a mathematician in Cambridge, England has crossed your path yet, but it is absolutely worth reading. The author does some outstanding investigative journalism into the pricing strategies that large publishers have been practicing. Perhaps if enough people read this work, then it will help to break the cartel formed by major publishers at the moment and push forward competitive pricing rather than the shielded by confidentiality clause type of unfair market that exists today, or better yet, a move towards all journals being open access. The publishers add almost nothing in a post-web world and yet wield so much power. The article is here," and then he gives a reference. "P.S. Love the show and keep up the good work. Dunk."
Yeah, this is quite a nice article about the publishing industry. Mm-hmm. It has a lot of numbers and arguments and so forth. So I would suggest you check it out. I'm not sure they add nothing post-web.
No, we don't want to put Alan out of a job.
That's right.
No, there are some things, but there certainly is a place for just articles somewhere. Yeah. Kathy, you're next.
Alice writes, "Dear Twivers, I was quite interested in the email from Randy, which you read on TWiV number 280. He became interested in virology after taking care of a stray dog who had canine distemper. I fell into it the same way. About 4 years ago, I stumbled on a Facebook group that was trying to find homes for dogs in the hellhole otherwise known as the Miami-Dade Animal Shelter. There was a pitiful dog covered with healed scars who was a pit bull lab mix. They were going to kill him if nobody claimed him that day. Something about him touched me, and I told the group I'd adopt him. But since I live in Laurel, Maryland, where it is partly cloudy and 63 degrees Fahrenheit, one of the group who lived in Fort Lauderdale offered to board the dog at her vet's until I could pick him up. I named him Jude. Jude had apparently been a bait dog for dog fights, hence the scars. I flew down to Florida to pick him up, but when I got there, I was told he was spiking fever, and they determined it was distemper. The vet explained to me what distemper was, that it was a virus and so there was no antibiotic for it, and that you could treat the symptoms until it went neurologic, But if that happened, he would recommend that Jude be euthanized. I agreed to that. Jude was there for a couple of weeks, and since he had barely escaped getting killed in the shelter, I was going to try to keep him alive to beat this, no expense spared. We were all optimistic, but it was all for naught. One day I got the call that he was stumbling around like he was drunk and suffering seizures. I tearfully told them to go ahead and let him pass. I had already begun reading everything I could find about distemper and other viruses, and I now have a particular interest in the morbilliviruses. I no longer snicker in Gone with the Wind when they showed that Captain Hamilton, spoiler alert, died of pneumonia following an attack of measles, which I used to think was just some routine childhood disease that only goofy Charles Hamilton could die of. I now know how extremely deadly that family of viruses can be. Please have Paul, the canine distemper guy, on. I think that would be a fabulous podcast, and maybe he can talk about the other viruses in the family. I'm especially interested in the morbilliviruses that are killing dolphins and other cetaceans. I've taken Vincent's courses, bought the book in capital letters. I read the blog in capital letters, and I listen to the podcast, also capital letters. Like Randy, I wish I had found these little buggers 40 years ago. I would have chucked law school. I did manage to get a nursing degree recently. There's that. I do have a question out of all this. Someone suggested that the distemper shot that Jude received when he left animal control might have made his illness worse if he already had the disease. I assume it's a live virus vaccination, so might it have made things worse for him? This is a totally academic question. Of course, he would not have been released without getting the shot. Love the show, Alice.
I don't know the answer to that.
I don't either.
The Baker Institute would know the answer.
Well, Paul Dupre also.
Right. And so the Paul she refers to is Paul Dupre, who you have now in the meantime had on, right?
Yeah, although we didn't talk much about canine distemper, I think, in dolphin morbilliviruses. I'm trying to see what the vaccine is. Is it an infectious vaccine or an inactivated one?
I'm looking at it.
So the question is, could the vaccine have made the illness worse, Dickson? So if you were infected and then
you've already got the virus,
so I don't see that it would do that.
I can't, unless it was given intracranially.
No, it's not given intracranially.
No, I know.
So I don't think that it's not likely that it had anything to do with the dog's progression. It seems to me.
Don't think so either.
But of course, we are not experts, and there's certainly vets listening. So
right.
You can weigh in. So, Alice, I hope you enjoyed Paul's podcast, which was not too long ago. And maybe we'll have him back to talk about dolphins. Alan, you are next.
Okay. Stephen writes, Dear TWiV Gunners, first, the term gunners is a term often used in medical school to refer to an individual who makes a habit of going above and beyond. It's part of their personality. It is a compliment. Second, I last wrote regarding the cyclospora epidemic in Iowa and Nebraska.
Oh, nice.
I'm from Iowa. You'll be happy to know we all survived and are waiting for the next epidemic.
And it's coming, believe me.
The real subject of my email is episode 282 in the discussion about Tamiflu and the subsequent discussion about research, funding, etc. I found much with which to agree in this discussion. A point I would make is that one does not get to translational research until one has done good basic and bench research, and I agree that not everything needs to have a clinical application. As a practicing physician who also has a strong interest in science beyond medicine, I think we have not done as good a job educating the public, physicians, and policymakers, as well as university administration, about how science functions. There, unfortunately, is the reality of commercial funding / influence in science, which can be especially apparent in pharmaceutical research. I don't have the answer. More public funding, a coalition of universities, an international approach, some other arrangement? Thanks for a great podcast, and keep up the good work. Just posted, but forgot 2 items. First, Garrison Keillor was the commencement speaker at my graduation from Luther College.
Nice.
He is, as you recall, the author and creator of A Prairie Home Companion with its chapter Lake Wobegon. Just a tidbit of trivia. Pick of the day / week, if you would consider How to Think Like Sherlock Holmes by Mariah Konokova. This is required reading for every student who comes through my practice and is a well-written book geared to the motivated amateur and easily readable by the professional in any number of disciplines. The ideas have broad applicability, and I can say that I'm a better physician since reading it. Again, thanks for a great podcast.
Look at that.
Cool.
Yeah.
Going back just a little bit, I finally found something about canine distemper vaccine. There's evidently a live distemper vaccine, and there's also a distemper vaccine that involves introducing a dog to a live recombinant virus that is not the distemper virus, that carries only a part of the distemper virus. So,
Either way, I don't think it would exacerbate
No.
the infection, right?
right.
Not likely.
Seems unlikely.
It does.
All right, the last one for today. Maka writes, "Hello, professors. This morning I was thinking that someone needed to tell Vincent that the state located between Washington and California is Oregon, not Oregon. Rhymes with sun. Oregon. Just a helpful hint." Okay. I got to remember that. Oregon.
Yeah.
It's like
Oregon.
Edinburgh. Okay.
Yeah, it is like Edinburgh.
Well,
Or
Melbourne.
Lozenji.
Melbourne.
Los Angeles, Dickson,
That's right.
Norwalk, Connecticut.
Norwalk, Connecticut. That's right. And Ohio.
Once in a while, a virus-related article will come through my horse channels, and I wanted to pass it along to you. Is there a channel about horses on TV?
I have no idea.
Just kidding. I think that she means other channels. Thanks for my weekly science dose, and now I have to run off and feed the horses. Really? So she sent an article from a magazine or journal that we often do not look at called The Horse, Your Guide to Equine Healthcare. Bornovirus antibodies detected in horses in Iceland.
Aha.
So bornovirus is an RNA virus that is known to infect horses and cause neurologic problems. And
Cool.
for the first time, its presence has been detected in horses in Iceland,
Wow.
which is quite isolated.
Which apparently does not allow horse importation. Interesting.
Yeah, so how it got there is quite the question, right?
It's quite the equestrian.
Dickson, Iceland is quite isolated, isn't it?
It certainly is.
Yeah.
Although it's not isolated in the sense that it receives a lot of air traffic, because it certainly does.
Yeah. So it might have
And disrupts a lot of air traffic occasionally.
Occasionally.
by the volcanoes.
Yeah.
Yeah.
Yes.
So Ian Lipkin played a major role in the discovery of this virus. Did you know that?
No.
He told me this story during a recent interview. Yeah. Perhaps we should talk about it sometime with him
Right.
if he's interested. All right, let's do some picks for the week. We do have more email here.
Right.
Oh, yeah.
Let me see. Let's get to the picks. Dixon, did you pick something this week?
I did. In fact, it was a very recent pick. I had lunch with someone who had worked in my lab slash office some time ago, and who was, in my view, so brilliant, a high school student, that at the end of his experience in working with me, I wrote him a glowing letter of reference to a northeastern school somewhere up in eastern Massachusetts, namely Amherst. And lo and behold, despite the letter, he got in. So now he's back in New York City to tell me what he's doing after graduation. And Kathy, you'll be very interested in this because he's working for an organization called Girl Rising. You ever heard of it?
No, I haven't.
It's the motto is "One girl with courage is a revolution," and it's about a group of people, a pro bono group of people. Actually, it's a 501c3 who devote their entire lives to encouraging the education of women around the world in various situations. So he's the campaign data engineer. Ben Pullman is the campaign data engineer. It's fresh out of college. This is his first job, and he spent half of the lunch hour telling me how exciting it was to work for this group because what he's really doing is helping them promote the idea of universal education. And they use 9 or 10 different examples from around the world. They've made a first-class Hollywood-level movie on this subject, and I highly recommend you going to their website and checking out what they do. So
This is cool.
That's my pick of the week. It's
Nice.
called Girl Rising.
Neat.
Very nice,
Indeed.
Dixon.
Thank you.
That was easy, huh?
And thank Ben.
Yeah, you go to lunch, you get a pick.
You got it.
Alan, what do you have?
I have a book that I've been reading off and on for a while, and I referred to it a little while ago, I think, but I have not picked it as far as I know. It's called The Science of Good Cooking. And this is it's kind of a cookbook. it's not really something to read cover to cover. I bought the Kindle edition, and I would probably recommend getting the hardcover instead, because it's the kind of thing you ought to just flip open when you're interested in a particular type of food or recipe and look at the recipe, and then it explains the science behind that recipe.
Is this a very expensive book by any chance?
No, it's $20.
Oh, okay.
So it's from an organization called America's Test Kitchen.
Oh, yeah, they're great. They're terrific.
They actually do the science behind cooking. That's their thing.
Yep.
They'll take a roast out of the oven and another identical roast out of the oven, and they'll let one of them sit for 30 minutes, and they'll carve the other one immediately, and then they'll measure the quantity of juices that come out of each, and they'll explain why you need to let a roast sit for 30 minutes after you take it out of the oven, and then they'll give you a delicious recipe for roast.
And then they get to eat it on the air, and you get to see how delicious it is.
Well, it's a book, but you can do these things yourself, and that's where I learned about the Maillard reaction. And all sorts of other interesting stuff about why particular things are done or should be done in particular ways in cooking something, and how all this stuff works.
So you probably won't be surprised that I've put this on my wish list. I own already at least 3, maybe 4 books about the science of cooking.
Nice.
I told this to one of my colleagues once, and she thought I was crazy.
You're not crazy. Have you ever heard of Alton Brown?
Oh, yeah.
Yeah.
Okay, he's got a program called Good Eats, and on several of them, he delved into the science of everything about, why things stick to pans, and, why the refrigerator is not such a great place to store something unless it's covered, and all this other stuff. And it's great to learn about the everyday way in which we live our lives with regards to the stuff that you put in your mouth. So, Alan, great pick.
You like to cook, Alan?
I do.
Me too.
Yeah, I do most of the day-to-day cooking. My wife usually does the cooking when we're having some big fancy meal. But when it's just time to make dinner, that's pretty much my job around here, and I really enjoy it.
Good.
Since I left the lab, I've got to fill that need somehow. I've talked to a lot of people who after leaving graduate school or postdocs, they went on to do something that was out of the lab, and there's this common theme that one of the things that you miss when you make that transition is the actual manipulation of stuff and setting things up. And cooking kind of captures a lot of that same thing, except with a lot lower risk of failure.
Cool. Kathy, what do you have?
I picked Daily Overview, and this is a site where every day they do a view of the Earth, and they think about where they want to get their photographs from. And so you can, when I saw the first one, I immediately subscribed to it, and I get it by email every day.
Cool.
So you can go back through the archive, and they focus on places where human activity has shaped the landscape. And so it's pretty cool. You see a train yard. There's a place, if you mouse over in the top blue part, you can get the top 10 or the archive, and so forth. And it's just, when it comes in the inbox every day, you just click on it, look at it for a minute, and I would say 80% of the time there's someone I know that would find this interesting because they lived there or something, and I forward it. So that's been fun.
It's cool. The one there right now is Washington, D.C. Neat. That's cool.
This is cool.
Very nice. My pick is the report of the investigation into the cause of the 1978 Birmingham smallpox occurrence, published in July of 1980. Now, this is a PDF, free PDF, which you can get from a link in the UK. And it is a report that was commissioned after the death of a medical photographer in 1978 who worked at the University of Birmingham. She acquired smallpox from the laboratory beneath the floor in which she worked. And she died. She spread it to her mother and father. They recovered. And the lab below her was working on the virus. This was just when it was about to be declared eradicated. And it's really amazing that this laboratory was working on the virus in what turned out to be not very good conditions. The PI of that lab eventually committed suicide. He was distraught over her death. And from this actually came a lot of the biocontainment procedures that we use today. In fact, this the reason I have this is because I had a meeting of the Institutional Biohazard Committee, of which I'm a member here this week, and it was a presentation about how this influenced current practices. And it's kind of, it's very interesting because you see how there were really poor conditions in this laboratory that probably led to the virus somehow getting into the air handling system and moving onto her floor and infecting her. Interesting thing, many interesting things about this. She had been immunized 12 years previously against smallpox, and apparently had not either taken or not lasted and so forth. So this is one of the incidents cited in that article that was mentioned at the beginning of the show, dangerous viruses escaping laboratories. But it's not really fair to cite that today, because today our security and containment and procedures are all so different because of this incident.
Sure.
Right.
And this would not happen again. So I think this is a worthwhile report to read. It's rather long and detailed, lots of photos. Yes.
There's a table of contents, and I misread it because actually at the first part, they actually refer you to the number of the paragraph. So I tried to jump to the conclusions, and I was in the middle of the appendices, and I had to go back and forth several times. So if you do that, notice whether you're looking at page numbers or paragraph numbers in the index or table of contents.
Vince, do you know in that article you just mentioned about how viruses have escaped from the laboratory, do they mention the Lassa fever incident?
What's that, Dickson?
Remember the one I told you about on the one of our early TWiVs?
Oh, so the guy getting infected at Yale.
Yeah.
Well, I think he or she mouth pipetted.
No.
There was a mouth pipetting infection.
But his technician caught it by an accidental needle stick.
Yeah, someone else mouth pipetted
and he went home and died.
Yeah, well, Dickson, that was again in the 60s.
I know.
When we didn't have much infection control
you're quite
procedures.
Right. You're absolutely right. So maybe they should have mentioned that as well. Because that was pretty dramatic.
Yeah. And I don't know where I'd find that, Dickson. I'd have to look it up.
It seemed like that Slate article cited a lot of things that were from a long time ago.
Yes,
Yeah.
they were mostly either from a long time ago or otherwise not relevant to the discussion.
Right.
That's kind of, that's the shtick there.
That is actually the book Fever by John Fuller.
Yes.
Has that in it, Dickson.
Yes,
That would be
yes.
a good thing. We talked about that on that episode.
We did.
Remember?
Do I remember?
That was like episode 2 or something.
Something like that, too.
That book got me interested in virology. Anyway, so that's the pick.
Uh-huh.
We had a listener pick already, and then there are more here. Let me just mention a few of them. This article is very slow scrolling because it's big. I don't think we reduced it very much today. Johnny, our physician friend from Cambridge, Massachusetts, writes another great website for introducing children to science. And this is called the RI Channel, the Smart Place for Science. Cutting-edge research, intelligent debate, and the leading thinkers in science, technology, math, and engineering. Lots of resources there. And you're not there, Dickson.
No, I don't think I ever will be either.
Ricardo writes, hello, Vincent and all the TWiV members. Listening as usual and getting richer and richer intellectually by the episode. This time I'm writing to send a listener pick. Gabriel Martens, the head of the Advanced Microscopy Unit at Instituto Gulbenkian de Ciencia, won the first prize on the Nikon Small World in Motion Competition 2013
Nice.
using a microscope that he built for $4,200.
Wow.
Then he published it in Nature Methods, Open Spin Microscopy, an open-source integrated microscopy platform. And he and his colleagues also created a do-it-yourself webpage so everyone can build one.
Cool.
It's pretty cool. Best regards and keep TWiVing. You are doing it very well. Thank you.
Well, we took TWiV lessons.
Yeah, they are. That's a journal. And Chris writes, hello, professors. For your consideration for a pick of the week, Gut Check, the microbiome game. You like that, Dickson? When you make that noise...
Yes, I like that, Vincent.
This is from Jonathan Eisen's lab. Jonathan Eisen,
yes.
microbiologist at University of California, Davis. And it is a game for 2 to 4 players that aims to strike a balance between learning some microbiology and having fun. You have to print out the cards, and each card has a pathogen, and there are points associated with getting certain ones. like C. diff, -2 points, and so forth. That's very cool. So you can get cards, rules, and player sheets and board at this website.
Cool.
This is great.
And because it's Jonathan Eisen, it's all open access.
Yep. And there's a note down below says we're planning to make this a drinking game. Okay. All right. Chris is a longtime TWiV, TWIP, and TWIM listener and epidemiology PhD student. All right, Laura, let me just write here that I'm ending the picks here so we don't go next week. And that will do it for this episode of TWiV number 289. You can find it at iTunes and twiv.tv. And do send us your email. We will get to it eventually. To twiv@twiv.tv. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is fun. It's good to get caught up on these, and the listeners come up with a lot of different things. It's always.
Don't they, though?
I'm always impressed. They're great.
They're
Yeah.
our extended family.
From the longest to the shortest, they're just great.
They're part of our TWIVome.
That's right. They are part of this excited and engaged community, Dickson.
There you go.
Alan Dove is at turbidplaque.com. You can also find him on Twitter. Thank you, Alan.
Always a pleasure.
And Dickson Despommier can be found at verticalfarm.com.
That Vincent just remodeled.
Should be up this weekend.
Oh, thank you.
By the time you hear this, the remodeled version should be up.
Did you add more verticals to the site?
Added more verticals. That's right.
More data.
Thank you, Dickson.
You're welcome.
I'm Vincent Racaniello, and you can find me at the blog, capital letters, virology.ws. I love the way she put it. That's great.
The blog. That's right.
And on your new website, we have the book and the podcast, Dickson.
That's right, Vincent.
You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.